Okay, let's go on here. Let's talk about membrane transport. Now, with membrane transport, that's movement of, in the cells, in the physiology, generally the movement of substances across the cell membrane. But let's go back. First of all, some definitions. Solution. A solution equals a solvent, which is the fluid portion, plus a solute. In your example up there, it shows sodium chloride, which is a salt or saline solution. Diffusion is defined as the random movement of a substance from an area of greater concentration to an area of less concentration. The illustration we would have used in the lab is a drop of food coloring in a glass of water. It moves and diffuses throughout the glass of water from a higher concentration where that food coloring was out through the entire glass of water. This is caused by Brownian movement, or the movement of the molecules and bumping into each other and pushing them throughout the glass of water. The other thing that happens is if you heat the water, the molecules move faster, and that causes the diffusion to speed up. Like I said, diffusion occurs in the constant movement of the molecules. Polarity. A solution or a solute that is polar has charged parts on the different ends of the molecule, while a nonpolar has no charged parts. A polar solution would be water. Polar solute would be sodium chloride. The nonpolar would be benzene or hexane or something along that. The cell wall is generally nonpolar, which causes it to, one, be degraded by nonpolar solvents, or have nonpolar solvents go right through the cell wall and lyse or kill the cell. Polar solvents, such as water can get in because it's small in size, but larger polar substances cannot. Or in some cases, the cell has aquaporins in it. The cell wall has aquaporins to allow water in. Now we're going to talk about the concentrations of the solute in the solvent in making a solution. There are a few terms. You have molality. Molality equals 1 mole of this substance, either a compound or just a single substance such as sodium. But a compound such as sodium chloride, the molecular weight is 58.5, and 1 mole solution is 1 mole of sodium chloride, which is 58.5 grams, plus 1 liter of water. Now you have molality, now you have molarity. Molarity is 1 mole of the solute, i.e. 58.5 grams of sodium chloride, with enough water that if you were in a graduated beaker to come up to 1 liter. So it's 1 mole of the substance and water 2s, or quantity sufficient to make 1 liter, which is a little bit less than water than a molal solution. Because the size of the compound changes, the molecular size, molarity is not quite as accurate as molality, where molality you have the ratio between the fluid and the compound being the same no matter what compound you're using. Now, osmosis. Osmosis is the movement of water or other solvent across a semi-permeable membrane from an area of low solute, thus high solvent concentration, to an area of high solute and low solvent, i.e., like we saw with the egg. The egg was in distilled water. The distilled water has no solute. By definition, it's distilled water. The egg has all the albumin and the other protein solutes inside the egg's membrane. Therefore, if you put the egg in distilled water, water is going to move into the egg as much as the membrane of the egg will allow. When it stretches as far as it's going to stretch, then osmosis ceases simply because of the membrane. If you have a weaker membrane, such as a red blood cell, as you saw in the lab when you put the red blood cell into distilled water, the water was able to go through the membrane into the red blood cell. The osmotic pressure, or the pressure to bring water in, was stronger than the red cell membrane, so the membrane ruptured and the cell disappeared. With the egg, the egg membrane was stronger than the osmotic pressure pulling the water into the egg, and so the egg did not rupture. It just grew very firm and hard, and it hit kind of an equilibrium. Okay, osmolality. Osmolality is measured in osmoles, and it is the pressure to pull water or solvent across that membrane via the change in concentration of a solute. So, if I have on one side of a membrane a 1 osmol sodium chloride solution, and I have on the other side of the membrane a 2 osmol sodium chloride solution, the water from the 1 osmol side is going to move across into the 2 osmol sides until the concentration or the ratio of solvent to solute is the same on each side. Then you'll just get some Brownian movement or some diffusion back and forth, but there will be no real net gain of solution on either side of the membrane. Now, another thing to keep in mind when you look at osmoles is you look at the ionization. If, for instance, we're using a compound that ionizes into 2 different compounds, if you take a 1 mol solution, whereas you have 1 mole of the compound plus 1 liter of water, when that compound is in the water, it dissolves, and it gives you 1 osmol for each part of the compound, i.e. sodium chloride. If I put 58.5 grams in 1 plus 1 liter of water, I come up with a 1 mole solution. However, once it's in there and it dissolves, the sodium gives 1 osmol because there's 1 mole of sodium, and the chloride gives 1 osmol because there's 1 mole of chloride. So you're going to have a 1 mole solution of sodium chloride, but it's going to be 2 osmoles. So conversely, if you have a solution like glucose that does not ionize, and you made a 1 mole solution of glucose, that would still equal 1 osmol because it doesn't ionize. Tonicity. Tonicity is relative to another solution. So if you have, going back to this sodium chloride solution, if you have a 2 molar solution on one side of the membrane and you have a 1 molar solution on the other side of the membrane, the 2 molar is hypertonic compared to the 1 molar, meaning hypertonic meaning it's more concentrated. The 1 molar solution is hypotonic compared to the 2 molar sodium chloride solution. Going on from there, say you take a 1 mol sodium chloride solution and you compare it to a 1 mol glucose solution, they're the same in molality, but in let's just go back. Let's just stay with the sodium chloride. The 2 mol is hypertonic to the 1 mol. The 1 mol is hypotonic to the 2 mol. If both sides are equal, then they're considered isotonic, meaning that there would be no flow of water back and forth. They're the same osmoles on either side. Now, another thing you're going to look at, and you're certainly going to see in the medical field, is a percent solution. And that is simply defined as the number of grams of solute in 100 milliliters of water. So a 5% glucose solution is 5 grams of glucose dissolved in 100 milliliters of water. That's Don't make it any harder than it is. That's a 5% solution. Normal body saline, normal body is a 0.9% sodium chloride. That's what a normal saline is if we hung it on a patient. That means there's 0.9 grams of sodium chloride in 100 milliliters of water. Now you may be asked to calculate the difference, or calculate between a molal solution and a percent solution. It's relatively easy. Percent, again, is in grams per 100 milliliters. Molal is in liters. So you're going to take the grams per 100 milliliters, multiply it by 10, and divide it by the gram atomic weight. And that will give you your molal solution. So for instance, if you have a 5.85% solution of sodium chloride, a 5.85, that's 5.85 grams of sodium chloride in 100 mils of water. To convert that to a molal, you're going to multiply that 5.85 times 10, which gives you 58.5, and you're going to divide that by the gram atomic weight of sodium chloride, which I actually did this on purpose, is 58.5. That will give you a 1 molal solution. So that's how you change, that's how you calculate the difference. Another factor that we look at, especially in the body for transport, and especially with the fats in that, are the detergents. Now the detergent bridges the polar and nonpolar, and we'll talk about that in a second. Actually, let's talk about that right now. We said that water is polar, sodium chloride is polar. That's because it has charged particles, whereas your fats and your hexanes and your benzenes, your organic compounds, are nonpolar. They don't have the charged particle. If we remember in the lab, when we mix them together, like oil and vinegar, they don't mix. They separate. When we added the detergent, however, they did mix. And the reason that happens is because the detergent has a polar and a nonpolar end. The polar end grabs onto the polar molecules, the nonpolar grabs onto the nonpolar molecules, and it binds them together. Now this is done by bile in the body. It's excreted by the gallbladder after a fatty meal. And what it does is it brings the nonpolar fat molecules or lipid molecules close enough to the polar enzymes so that the lipids can be broken down by the enzyme, digested, and the glucose and the energy absorbed for body function. So that's nonpolar. That's how detergents work. You know, it's just like Joy or Palmolive, whatever detergent you have in your sink. It grabs the grease off the dish, binds it with the water, and you wash it down the drain. In the human body, it's the bile, and the main one is bile. It grabs the fatty area, the fat molecule, and then the enzyme brings them closer together, allows the enzyme to work on the fat molecule.